welcome. This is going to be part 2 of chapter 15, focusing on our chemoreceptor senses, what we collectively refer to as the chemical senses, which is olfaction and gustation. So olfaction, of course, is smell, gustation is taste, and there's really two reasons that they get studied together. One is that they both use chemoreceptors that carry out the transduction, and the senses are kind of complementary. They do go hand in hand, and kind of the evolutionary advantage to our chemical senses is it's really about knowing which foods are foods that we should eat because they would contain nutrients or vitamins that would be good for us, and which foods we shouldn't eat because they could potentially be harmful to us. So yes, these are chemoreceptors. The receptor cells for olfaction are dissolved in kind of the nasal fluids that are kind of in the your mucousy nose there, and then the taste receptors, those chemicals are going to be dissolved in the saliva. In order for our chemoreceptors to be activated, whatever chemical that they are responding to must be dissolved in some sort of aqueous solution, otherwise they won't work. That's why when you guys did hopefully did the test with the sugar on your tongue. If you dry your tongue and put sugar on your dry tongue, it doesn't taste right away. You can't taste the sugar right away because the sugar can't dissolve on your dry tongue. You need to have some saliva there for it to dissolve it. And once you have, your kind of tongue gets re-wet by the saliva in your mouth, then you can taste the sugar. And that's why typically it takes a little bit when you do that particular test. So let's talk about olfaction first. When we refer to the word odorant, what we are talking about is whatever chemical happens to be causing the odor. Odorants, in order for, again, because our olfactory receptors only work and only respond to chemicals dissolved in fluid, in order for us to smell a specific odorant, that odorant must be volatile, which means it must be in a gaseous state, because if it's in a gaseous state, it can then dissolve in the fluid surrounding our olfactory epithelium. What you think of as a specific smell or odor typically has several different odorants in it. It's not just one chemical that you are smelling. It's kind of an integration of all these different chemicals. We can distinguish about 10,000 odors, although that's not nearly as good as other animals. We have about 400 genes that only become active in the nose. Each of these genes encodes for a specific receptor protein that will respond to usually one or more of these odorant chemicals. Most odorants bind to several different receptors, and it's kind of the combination of these, which receptors get activated by which odorants that are really responsible for what we think of as smell. Like I said, we have about 400 smell genes, and we can taste or smell, about 10,000 odors. I think like dogs, which have a much better sense of smell than we do, have something like, 10,000 smell genes. So just imagine how many odors they can smell with that many genes. Certainly a whole lot more than we can. When we talk about the transduction of smell, this is of course happening in the olfactory epithelium, which is on the roof of your mouth. And here would be our little receptor protein that's going to respond to this specific odorant. This is a fairly simple process, kind of very normal transduction process, completely unlike vision, which is much more complicated. In the transduction of odorants into neural signals, chemicals to neural signals, it uses the G protein process. So here we have our odorant. It binds on to the receptor protein. That receptor protein goes and activates the G protein. The G protein goes and activates adenylate cyclase, which is another transmembrane protein there. Adenylate cyclase then produces a second messenger called cyclic AMP. Cyclic AMP then goes and opens up a cation channel so that both sodium and actually calcium come into the cell of the olfactory epithelium, and then it gets depolarized. And if we depolarize to threshold, then we get an action potential. So here we are in the olfactory epithelium at the roof of the mouth. Here's the ethmoid bone. You can see here's the foramina and the cribriform plate. And you can see here's our olfactory nerves connecting to the receptor cells here. So the receptor cells here into the olfactory bulb, into these little kind of webs that we call glomeruli. So the olfactory epithelial cell here, this bipolar sensory neuron, is the first order neuron. It then converges here in the glomeruli of the olfactory bulb, where it connects then to the second order neurons, which are the mitral cells. And then the mitral cells are what combine and merge into the olfactory tracts, which take it then into the brain. Let's talk about these glomeruli here. You'll see that it's multiple receptor cells kind of converging. They're going to all respond to a specific odorant, or at least with the same receptor type. Then the second order neurons, the mitral cells, then kind of amp up that response and really only send the really highly excitatory signals into the brain. So if there's not really enough of that particular odorant, that might not get included in the kind of congruence of smells that make up a specific odor or smell. The mitral cells, their axons, as I said, merge into the olfactory tract, and then the olfactory tract goes right to the olfactory cortex of your brain. Unlike every other sense, with the exception of some of the proprioception that uses the spinocerebellar pathway, this is the only sense that does not go to the thalamus. All of your somatosensation, all of your special senses except smell go to the thalamus first, have their second order neurons or first order neurons that take them to the thalamus. The exception to here is this olfaction. The olfactory tract completely bypasses the thalamus and goes right from the olfactory bulb to the olfactory cortex of your cerebrum. Some of that kind of smell information also goes to the prefrontal cortex so that you can consciously identify, hey, I'm smelling roses, cookies, take your pick, skunks, something noxious. And it also sends some of that smell information to things like the limbic system and the mammillary body and the hypothalamus, so that we get kind of those emotional responses to smell. In fact, smell is one of our strongest triggers of memory, which of course can be very much limbic system and hippocampus. So let's talk about taste. When we talk about taste, we're talking about the taste buds here on these little papillae of your tongue. Each of these little hair cells are either going to be some sort of kind of actually the actual gustatory cell or some stem cells. And let me go back here for a second because I didn't mention that here. You'll also see lots of supporting cells in the olfactory epithelium. Both our olfactory cells and our gustatory cells have some stem cells that divide all the time and can be replaced quite easily because these gustatory cells, and actually the olfactory cells as well, can get damaged fairly easily. And so we want to be able to kind of replace them if they do get damaged. So they actually regenerate much easier than most other neural cells. We have 5 basic cells, 5 basic tastes that our body responds to. We have sweet, sour, salty, bitter, and umami. When they all respond to a specific chemical. So sweet is basically responding typically to sugar. Sour is responding to acidic things like hydrogen ions. Salty is usually those, well, shockingly enough, inorganic salts like sodium chloride, metal ions. Bitter is responding to alkaloids. We find them in quinine and nicotine. We find alkaloids in aspirin. That's why if you leave an aspirin on your mouth and your tongue too long, it's like disgusting. That's because it's that bitter sensation. And then umami is actually a Japanese word. And the umami cells respond to amino acids, 2 particular amino acids, glutamate and aspartate, but protein, or the building blocks of proteins, amino acids. We do suspect that we actually have a 6th taste as well, outside of our 5 basic tastes, and those receptor cells would respond to fatty acids. So this may somewhat explain why we very often like things that are not so good for us and that are fatty, because maybe we have developed a taste for that. The idea behind our different tastes is that they're essentially telling us and driving us to go eat the foods that we should be eating and avoiding things that we should not be eating. So sweet responds to glucose. Well, glucose is the molecule by which we make energy. That's important. We need energy. Salty, and we're going to go crave things full of sodium chloride. Well, sodium is incredibly vital to the functioning of your nervous cells. If we look at umami, that's that nice meaty, proteiny taste that is so yummy in meat and mushrooms. And it's because our body requires amino acids to make proteins. Proteins do everything in our body practically. And so we're driven to seek out food by our tastes that contain things that will be useful to our body in terms of nutrients. Now with bitter, most bitter foods, or a good kind of rule of thumb, is things that generally taste bitter could potentially be toxic. And so if we put something in our mouth and it's bitter, we spit it out right away. And so we're not going to ingest something that's harmful to us. And so that's why we've evolved these tastes over time. It's not just to make our food taste good. Of course, it does do that as well. But there is an evolutionary advantage, a homeostatic value to our tastes. In terms of how taste transduction works, it's actually fairly similar to the olfactory sensation, in that the chemicals must be dissolved in some sort of fluid. In the case of gustation, it's the saliva in your mouth. The chemicals then dissolve into the taste pore and get actually into contact with the gustatory cells of the taste bud. That then depolarizes that particular gustatory cell, and it releases some neurotransmitter. That neurotransmitter gets released to one of our gustatory nerves, and we get a graded potential. And if the graded potential is high enough, if enough neurotransmitter gets released, then we get an action potential. Different tastes have different kind of thresholds. It's not always that typical action potential threshold. The bitter receptors are actually the most sensitive, is the easiest to activate a bitter receptor. Again, this is a homeostatic advantage. If you put something in your mouth and immediately tastes disgusting and you spit it out, you're less likely to have more of that potentially toxic thing in your body. So that's why our bitter receptors tend to be the most sensitive, because it's driving us to avoid something that could be harmful to us. There are both kind of direct depolarization and indirect depolarization in terms of the transduction of a chemical into an action potential in terms of taste. Salty and sour, since they respond to cations, salty to sodium and sour to hydrogen. These are both direct depolarizing events. The cation channel opens, sodium influxes, causes depolarization into a salty receptor. A cation channel opens, hydrogen comes in into a sour receptor. And then the serotonin neurotransmitter goes in synapses, released by the salty and sour receptors to our gustatory neurons. Sweet, bitter, and umami, so sugars, alkaloids, amino acids, they use that G protein kind of relay race, more indirect method. So if we were to go back, sweet, bitter, and umami kind of follow this pathway where we get eventually the release of some calcium. Salty and sour are just directly opening the cation channels. So direct depolarization, more of an indirect depolarization when the calcium comes in. In terms of our gustatory pathway, it starts here with our taste buds. They connect then to our two main gustatory nerves, the facial nerve and the glossopharyngeal nerve. So there's your first order neuron. That then synapses here in the medulla with the second order neuron. The second order neuron takes it to the thalamus, and then from the thalamus we get a third order neuron going out to the gustatory cortex in the insula. Very similar here with the vagus nerve, cranial nerve number 10. You do have some taste receptors kind of lower in your throat, but it's still the same process of first order neuron going from the receptor to the brainstem, second order neuron going from the brainstem to the thalamus, and then the third order neuron going from the thalamus to the gustatory cortex. Not only does it go to the gustatory cortex, but it will also go to the hypothalamus and the limbic system. That's where we get kind of that emotional investment in our food. And then also, again, as well to the prefrontal cortex, so we can consciously identify, hey, I'm tasting cookies, or I'm tasting, I had steak for dinner tonight, so many of my umami receptors were being activated by my steak. Taste is also highly influenced by smell. That's why if you're sick and you're congested, your food doesn't taste as good, because that taste, that lack of kind of smell component kind of tamps down on your taste. Thermoreceptors, so like the temperature of your food, right? If it's too hot or too cold, it's harder to taste. The mechanoreceptors, so kind of the feel of the food. if you go to like a hoity-toity restaurant, they're always talking about mouth feel. How does it feel in your mouth? But it's the idea of texture, you know. My brother hates beans because he doesn't like the texture of the beans. I can't stand asparagus when they're mushy, but if they're not mushy, they're great. And then actually nociceptors, the pain receptors. If you're one of those people who likes crazy hot spicy food, then you're kind of a masochist, because really 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 spicy food actually activates the pain receptors in your mouth, the nociceptors, which is kind of interesting, I think. I went out one time with a buddy of mine, and he ate those really. We were at a Thai food restaurant, and it was those green peppers that are super super spicy in Thai food, and he thought they were green beans, and he just piled them up on his fork and put it in his mouth, and I mean, you should just see his face. It was ridiculous. He was in so much pain. Needless to say, he didn't eat quite that many at one time again, but it was kind of fun. So taste is, again, smell. It's about driving us to eat the foods that have things that we need, like glucose, like amino acids, and avoid things that could potentially be toxic to us. So that's the end of our chemical senses.